So here we have the dreadful Ebola virus. So the general structure of the Ebola virus is this kind of long, elongated strand, which is all this capsid, which is made of proteins. And it's kind of hard to see on the video here, but I made one strand of red. That is the RNA inside the virus. And here we have the polymerase, which will become more important later in the explanation. So essentially what we have here is this DNA, right, that's covered in these proteins that give it kind of this structure and this protection from whatever environment it's in. And then what's really key is these glycoproteins, which are the little lock and key mechanism that the Ebola fits into when it hits a cell. It will fit into a certain receptor, and the cell will gobble it up like it was a certain nutrient or protein or something it was looking for. So here is kind of a more in-depth explanation of Ebola infecting a monocyte, which is one of the cell types that it infects, which is kind of one of the early immune cells that could respond to an infection. So when it finds a monocyte, it's going to find this specific receptor called this NPC1 protein receptor, which is a cholesterol receptor. It's kind of unclear. I found some conflicting opinions, but I think the more key thing that it does is once it gets inside, it binds to the NPC1 receptor in the lysosome, which is this little capsule that carries the virus through the cell. So once it gets here, it's going to bind, which I have a little better drawing here. So now it's in the lysosome in this monocyte in the cell, right? And it wants to get out into this area. This is the important place that it needs its RNA to be so that the machinery of the cell and the ribosomes will start to replicate it. So what it does is has this polymerase, which if you remember from the first page, kind of at the head of the virus here, there's these things that are essentially going to run the virus through and copy it out, as we learned in class. This polymerase is essentially the NPC1, the end of the Ebola will bind there and fuse with the kind of outer side of the lysosome, and it will start to kind of shoot out its RNA into the cell. And so it's important because in studies that they've done, if the NPC1 was not around, this lysosome will eventually kind of dissolve and start to destroy the Ebola virus. So the Ebola virus needs to take advantage of that while it can. And so what this kind of results in, right, is as that one cell gets overridden, it eventually will lyse and kind of shoot out more of the viruses, which will find more receptors. And so kind of the first casualties in this are the monocytes and the macrophages, which are the immune cells. And then it kind of moves on to other things which end up producing the symptoms of the disease. So it goes to nerve cells, dendritic cells, and liver cells. But most importantly for its presentation, it goes to epithelial cells, which are kind of these structural connective cells in the body. So epithelial cells you have in the base of your skin layer, but importantly in the case of Ebola, you have epithelial cells in your blood vessels. So what happens here is when these cells essentially get destroyed, you kind of eventually get these breaks in the blood vessels, hence the hemorrhagic fever. So as it kind of wreaks havoc in these epithelial cells in your blood vessels, fluids and blood start to kind of leak out into the body and the conjunctiva and everything, and so it causes a problem there. And so another key thing that Ebola does is it masks immune response and interferon, and so as all these cells are destroyed, the immune response is minimal until it kind of catches wind of it a bit later with other processes. But that's kind of the reason why it's especially dangerous, is it can cause a lot of damage before the immune response is great enough to start to handle it.